All right, so I think we'll go ahead and get started now. Welcome and thank you for joining us for today's webinar, Understanding Insecticide Resistance. My name is Jennifer Wells, Project Coordinator here at the National Environmental Health Association, and I will be your host for today. The webinar today is the second part of a two-part series. Today's topic is the importance of understanding resistance mechanisms and management theories. So to begin today's webinar, I'll highlight a partnership between two organizations and provide a brief overview of each and what they do. Then I'll introduce today's guest speaker, who has an amazing presentation planned for you. And we'll also have a Q&A session following the presentation. And then when we come to the end, we'll provide a few final announcements and then some upcoming opportunities. Now just a few housekeeping items as we get started. Please note that this webinar is being recorded. The recording will be uploaded onto the NEHA Vectors and Pests webinars page. You can also find recordings of previous webinars there as well. The web address is neha.org/vector-webinars. And then if you have any questions for our speaker during the presentation, please use the Q&A box to submit your questions and we'll get to them after the presentation. And you can find the Q&A box at the bottom of your screen. At the conclusion of the webinar, you'll receive a link to take part in an evaluation survey to provide feedback and comments on today's webinar. We ask that you please take a few minutes just to let us know how we can continue to meet your needs as an organization. So this webinar is brought to you in part through a collaboration between Gulf South Vector and NEHA through grant funding from the Centers of Disease Control and Prevention. So now I'll share some information about both organizations. The mission at the Gulf South Vector Educational Center for Training, Outreach, and Resources, or VECTOR for short, is to forge a robust, innovative, multidisciplinary, and cooperative regional network. They aim to provide innovative training and resources to professionals and pre-service students, creating a unified workforce that enhances regional capacity to monitor and respond to vector-borne disease threats. So if you want to know more about this organization, you can either scan the QR code on the screen or visit their webpage at vectoroutreach.org. And now for those who may not be familiar with NEHA, we support the environmental public health workforce. As an association, everything we do is to build, sustain, and empower environmental public health professionals so that they're equipped and prepared to protect the health of communities across the country. We support environmental public health by continuously providing the most up-to-date training, tools, and resources to the workforce who specialize in so many of the different fields considered in environmental public health. So NEHA is focused on increasing knowledge and skills across all our fields to build upon our workforce capacity, efficiency, and effectiveness. And we also offer credentials to certify the expertise of our workforce. In addition to our large online library of trainings, webinars, tools, and resources, we also host an annual educational conference, which includes multiple sessions focused on the many different fields of environmental public health, such as vectors, food safety, private wells, water quality, climate change, and so many more. And now, no matter where you are in your educational journey or professional career, NEHA has something for you. So please check out our website at neha.org/membership to learn more. And I do want to highlight that if you are a student, currently membership is free. So as I mentioned, each year NEHA hosts our annual educational conference and exhibition. Our conference this year will be in Phoenix, Arizona from July 14th to 17th and promises to be your oasis in the desert. Registration is currently open and we highly encourage you to attend. And you can learn more and register at neha.org/AEC or scan the QR code on the slide. All right, now let's get to our guest speaker. Please welcome Janet McAllister. Janet is a board-certified medical entomologist who earned her undergraduate and master's degrees from Louisiana State University. Her PhD in medical entomology comes from the University of Arkansas. After 20 years of public service as a research entomologist with the Centers for Disease Control and Prevention Division of Vector-Borne Diseases, she is now the principal research entomologist for the City of New Orleans Mosquito, Termite, and Rodent Control Board, where she supervises the mosquito control unit. She is an American Mosquito Control Association Medal of Honor recipient for outstanding contributions to the science and control of mosquitoes, other vectors, and vector-borne diseases. So with that, Janet, the floor is all yours.
Okay, just give me 1 second to share my slides. And it looks like you are seeing my screen, I hope.
Yes.
Okay, so as indicated, this is the second of a 2-part webinar series, and I'm going to flip the title a little bit and talk about management and then the mechanisms. All right. Screen sharing, there we go. So in the first part of the series, I gave a definition of insecticide resistance. I actually gave a couple of definitions because there are a lot out there. This is the definition that I like and use because it's proactive, and it's about resistance being a genetic change that may impair control in the field. Because I like this one because it allows you to actually recognize that you have resistance developing and that you can make changes in what you're doing to hopefully reverse that trend or to regain susceptibility before you have lost the use of products. I also talked about a bunch of assays that can be used to detect insecticide resistance. The time mortality assay, which is the CDC bottle bioassay. And if you are a program, you can go online at CDC and actually get free kits that include just about everything you need to run those assays. And I also talked about different nuances in dose mortality type assays, which are perfectly valid assays to be using in a resistance program or a resistance monitoring program. Okay. And finally, I talked about how a resistance monitoring program is really taking a series of snapshots and looking for trends over time, and not just a survey periodically done to see what kind of resistance may be present in an area. So today I want to talk a little bit about the different management theories for resistance, and they're listed here: moderation, saturation, and multiple attack. So management by moderation is just like it says, it's using less insecticide, lowering doses and numbers of applications so that you're not putting constant pressure on an insect population, that you're not treating all areas where the pest occurs, where you're using local treatments. And the bottom bullet point there is really more for agriculture, because we don't have economic thresholds in public health. But most mosquito control programs do have some sort of treatment threshold where they're going to, once mosquito numbers reach that a certain level, they're going to do spray missions or not. And that threshold in public health can change depending on whether there is disease present in an area or not. So management by saturation is really about trying to put out an overwhelming amount of insecticides so that those individuals that have these resistance genes are still overwhelmed by the products that you're using, either by using higher doses or by the use of synergists to enhance how well a particular product is going to work. There are a couple of assumptions for this, and that's the genes are dominant, but also that compounds have short residuals. And that for this to work, you really need refugia and immigration, where you have a source of susceptible genes to move back into the area so that the predominant population has susceptible genes in it. And then there's management by multiple attack, and this is using mixtures of chemicals or alternatively rotations or mosaics. I'll mention what those are. And there are some assumptions for this strategy as well, in that there's no initial resistance and that the mechanisms are different, and decay rates in the chemicals that you're using are different in the field. And again, some of that terminology in those assumptions is really geared more toward agriculture pests than public health pests, but the strategy is the same. So what does this look like in like more concrete? So if the two big squares that you're seeing are really the same area, so say that's your county or your town or whatever your jurisdiction is where you are trying to control, say, mosquitoes, and most places are going to subdivide their county or their town into manageable operational zones where they do surveillance in the different zones so they can tell what's going on throughout this large landscape. And say that you are using chemical X, which is represented by light blue in this case at a mid-label rate, and you determine that you have insecticide resistance. Well, one of your strategies could be to stop spraying and rely on other control methods. But the reality is that's really not that feasible, that you would just stop spraying all chemicals, because chemicals are a very important tool for public health. But that would be a valid strategy, is to stop spraying for a while and let those mosquitoes recover. Or you could put that light chemical X out at, instead of the mid-label rate, the highest label rate. And so you could increase the dosage, which is an example of using that strategy of management by saturation. Or you could change chemicals. And so this is more of that management by multiple attack, where you've been using chemical X, you're going to change to chemical Z. And you can make an overall change where your program is now just using chemical Z, or you could do a strategy where you are going to rotate between using chemical X and chemical Z. And that rotation frequency could be, I know programs that do that once a month. So in an operational zone, if the month of July they used chemical X, in the month of August and they needed to spray, they would use chemical Y. That time frame could be on a yearly basis, or it could be periodical, like perhaps every 3 years, where you would rotate chemicals. Or you could start doing a management scheme where you are using what's called a mosaic. So in some areas of your county or your city, you may use one chemical Z, and in other areas you may use chemical X. This has actually been demonstrated to be effective in malaria control programs, usually at a village level, where some huts might be sprayed with one chemical and other huts within the village would be sprayed with another chemical, to try and minimize the development of resistance. But the reality is that you may have to rely on more than one method. And so in your jurisdiction, you may have areas represented by white where you don't treat at all. This could be wildlife refuges that are off limits. They could be industrial areas where you really don't have a mosquito problem. You can have areas where your surveillance shows that you do have resistance, which is represented by the R's, in other areas where the product that you're using is working just fine and there's no detectable resistance. And so in some areas, simply increasing the label rate to the highest label rate may be sufficient to manage that resistance. In other places, you may need to switch. And in other areas, you may need to do some sort of a more frequent rotation as well. So how do you know which one of these strategies is going to be the best strategy to use when resistance is detected? And that's where understanding what the mechanism is can give you some insight. So if you remember from the previous webinar, there's a pathway that insecticides take to get to the target site and interact with that target site. And really, resistance mechanisms are places where that pathway is interrupted either through increased detoxification, which leads to increased excretion, where there's reduced penetration, where the mosquito is not absorbing the insecticide at a fast rate, so that detoxification can take care of that insecticide before it affects that target site, or you could have changes in the target site itself. So if you'll remember, I mentioned cross resistance, where you have a common mechanism that gives you resistance to more than one chemical, or you could have multiple resistance, where you have multiple mechanisms that can give you resistance to multiple chemicals. And so typically, you can have another type of resistance called behavioral resistance, which the insect changes its behavior so that it is no longer coming into contact with the insecticide to begin with. Reduced penetration and metabolic resistance mechanisms tend to be linked, but could certainly be standalone mechanisms. And altered target site resistance tends to that's if you have an altered target site resistance mechanism, it's only going to affect insecticides that work at that same target site. So it tends to give you resistance just to other insecticides in the same class. So altered target site testing, this is shows you two of the target sites that are targeted by public health pesticides. The pyrethroids and DDT affect the sodium channel, and then organophosphates and carbamates are going to affect neurochemicals that are in the synapse between to nerve cells. And so the pyrethroids, they affect the sodium channel, and the best way to detect sodium channel mutations is with PCR. You can also do it with phenotypic assays because this sodium channel mutation is what is called KDR, or knockdown resistance. And this is where the insecticide no longer binds effectively at the sodium, or no longer binds effectively at the sodium channel. And they call it knockdown resistance because phenotypically the insect is knocked down but recovers quickly. And so if you expose your insects to your chemical, your insecticide, and hold them for 12 to 24 hours and they recover, or a portion of the population recovers, that gives you an indication that you have KDR resistance, but that is only with pyrethroids. If you were to do this with an organophosphate, you would not see that same effect because the organophosphates are working in that nerve synapse. And so there is an enzyme assay, which I'll mention just briefly how that works a little bit later. Or you can do molecular tests, PCR, to look for the actual mutation in the gene that is giving you resistance to the organophosphates through an altered target site. The detoxification defense enzymes fall into three classes of enzymes. These are proteins, they are oxidases, esterases, and glutathione S transferase. And these are really a suite of genes. So they are a class of enzymes. When I talk about oxidases, for example, in Aedes aegypti, there are over 160 P450 oxidase genes. So there are a lot of these oxidases that are in Aedes aegypti. And which ones out of that 160 are going to give you insecticide resistance is not well characterized. There are certain ones that keep rising to the surface of, oh, these are overexpressed in individuals that may be exposed to, again, chemical X, but if you expose them to chemical Z, it may be different genes that are involved are overexpressed. And so while we talk about oxidase as a mechanism, just keep in mind that there are a lot of different oxidases, and that oxidase can be specific, that is overexpressed, and can be specific to one chemical, one pyrethroid, or one organophosphate, or they could be more general and give you resistance to a couple of pyrethroids or a couple of organophosphates, or even more general, and all of the organophosphates and maybe some of the pyrethroids may be affected by the particular P450. And the same concept is true with the esterases and the glutathione S transferases. These are multiple enzymes that an insect is going to have genes for and produce. So the insecticides that we work with, this is an example of a pyrethroid, but an organophosphate has a similar situation where you have an ester linkage, and you have a carboxylic end of that insecticide and an alcohol end of that insecticide. And then what makes the difference between the different pyrethroids is what is going on either side of that ester linkage. So some of them may add some chlorines and there are some bromines to make them a different insecticide, but they have the same general shape. And that's how we classify the insecticides, is by this basic backbone. There is one exception when it comes to the pyrethroids, which is why I like to use them as the example, and that is edofenprox does not have an ester linkage in it, and that's very important when it comes to perhaps managing for resistance. So if you look at that basic backbone, there are a lot of places on the general molecule that are open to attack by these enzymes. If there is an ester linkage, that is where an esterase is going to be able to attack the molecule. And then there are different points on the ends of it that can be oxidized. And the basic effect of the enzyme on an insecticide is that it breaks it into smaller molecules. It cleaves that ester. If you have esterases present, they're cleaving that ester bond, or they're oxidizing at different places so that you have a more water soluble littler chunks that can be excreted or sequestered. And so the insecticides that are affected by these different metabolic mechanisms, as you can see here, esterases can work on both pyrethroids and organophosphates. Oxidases work on pyrethroids, organophosphates. You can have overexpression of glutathione S-transferases that can work on pyrethroids and organophosphates. So our main workhorses for public health in the United States, which are pyrethroids and organophosphates, can be attacked by any of the three enzyme mechanisms that could be present. So how do you detect which one of these enzymes is important in resistance? Is once you have detected resistance, you can then use another chemical to basically wipe out the different enzymes and see what happens. So you have a population of, and this is a picture of Aedes albopictus. So you have a population of albopictus that has resistance to it. And you prepare a bottle with what we call an enzyme inhibitor. You expose the mosquitoes to that inhibitor by putting them in a bottle for about an hour, and then you take them out of the bottle and you let them recover from that exposure, but you also are giving them time for that inhibitor to wipe out one of the enzyme classes. And then you run the bottle test or your dose mortality test with just the insecticide where you determined there was resistance to. And you have one of three scenarios that can happen. In scenario A, that resistant population which is depicted in, it can go back to looking susceptible, and that tells you that enzyme class is phenotypically responsible or contributing to your resistance as the mechanism. You can have B, where it still looks just as resistant as it did in your population without the inhibitor, or C, where it goes back toward looking susceptible, but it doesn't go fully back to looking susceptible, which tells you you have that as a mechanism, but you have another mechanism that is also contributing to the resistance that you are seeing. So those enzyme inhibitors, and if you were to order the CDC bottle kit, you have the option of ordering these inhibitors as well. It's PBO that inhibits oxidase activity, SSS tributyl phosphorotrithioate, which is abbreviated as DEF, inhibits esterase activity, and diethyl malate inhibits glutathione S transferase activity. Now, PBO you may be familiar with because it is often put into products to synergize them, to have this multiple attack going on. But PBO by itself can be toxic to insects because it can wipe out all of their oxidases. Another way to measure which of these enzymes might be present is to use what are called microplate assays or biochemical assays. And in these assays, you grind the individual mosquitoes into with a buffer in a tube, and then you will add a substrate that is going to bind to the enzyme class that you are interested in. Such as using alpha-naphthol acetate to find alpha esterases. And then after you have added the substrate, which is going to bind to the enzymes that are present, you add an indicator chemical that would change the color so that you can get a measure for how much of the enzyme is actually present in your individual mosquito. Now, these are run on microplates typically, and so you can run, and we usually would run them in triplicate, so you could put 30 mosquitoes onto a microplate and test them at once. And you can read the microplate with a reader, and that's going to give you an absolute value. But if you don't have a microplate reader, you could actually read them by eye and get a relative value of if it's really dark, you have a lot of the enzyme. If it's really light, you don't, and you would have some controls on that plate as a comparison. And then finally, we do have one of these microplate assays, as I mentioned before, that can be used to look for the altered acetylcholine esterase mechanism, which is the altered target site mechanism for the organophosphates and the carbamates. And this just adds an extra little chemical into the mix that is going to compete with the substrate to bind the acetylcholine esterase that is giving you resistance as an altered target site. And so these biochemical assays are available for oxidase mechanisms, glutathione S transferase, non-specific both alpha and beta esterases, the insensitive acetylcholine, which is a target site, and then protein, because you want to know the size of the mosquito, because if it's a really big mosquito, it's naturally going to make more of these enzymes than a really little mosquito. And so you can use a protein assay that measures overall protein, and you can make a correction for size so that you know if they have a lot of one of these enzymes, it's not just because they were a really big mosquito. And you can, with this microplate format, you can actually run all of those enzyme assays on an individual mosquito. And so this picture here has the insensitive acetylcholine assay for the altered target site, oxidase assay, esterase assay, and the first three wells of each color across the top are the same mosquito run in triplicate. So you can see that first mosquito on the top actually has all three mechanisms in it, whereas the fourth mosquito down has a lot of esterase. Those three wells are really dark, but it doesn't have the insensitive acetylcholine esterases or the oxidases being overexpressed. So it's a really handy assay method that is actually pretty cheap. It's a lot less expensive than doing molecular tests because these chemicals to do these assays are not very expensive. A couple more things to consider when you are looking at enzyme assays is that some of these enzymes have different effects or expression levels based on the age of the mosquitoes. So with the example of oxidases, age does not have an influence on it. So the left panel are actual measurements of oxidases using microplates. The panel on the right in these next couple slides are bottle bioassay data for the same group of mosquitoes. And the overall esterase levels do not change whether it's a 3-day-old mosquito you're testing or a 17-day-old mosquito. And you can see that with phenotypically expressed in the bottle assay, where the 3-day-old adults were resistant and the 15-day-old result mosquitoes have roughly the same resistance level as well. But that's not the case when it comes to esterases. And so the age of the mosquito does have a large influence, and an old mosquito or old insect is not going to express as much esterase as it did when it's younger. In fact, the life stage where the most in a mosquito, the most esterases are expressed is actually in the pupal stage, where all of this rearrangement of tissue is going on. And so once they emerge as an adult, they slowly use up their esterases and they're not replenished at a great rate. And part of that is because what esterases do is they tend to not just break apart an insecticide, but they will also sequester it. So they will bind to it, and then the insect will store it in fat or be able to excrete it. But the bottom line is that a young mosquito may look resistant, but an old mosquito goes back to expressing susceptibility. And that's very important when it comes to disease transmission, because when a mosquito takes a blood meal, even if it's the very first blood meal, and becomes infected with a pathogen, it takes 10 to 14 days for them to be able to transmit to another host. And so if esterases are your resistance mechanism, and you really don't have a lot of options of chemicals to switch to, that gives you a little bit of boost or benefit to have an esterase mechanism over an oxidase mechanism in your population, because it's the old infected mosquitoes that you really need to control during an outbreak. So any one or combination of these mechanisms that I've talked about can be present in a mosquito population. And so you can have individuals that may have elevated esterases and other individuals with elevated oxidases, or you can have an individual that has both elevated esterases and oxidases, and maybe some altered target site mixed into the population as well. And so it becomes very complex very, very quickly on biologically what is going on in a population of mosquitoes when you detect insecticide resistance. And just to drive this home a little bit, this is some data from Anopheles albuminus, and you are looking at microplate data. where the esterase activity is along the bottom and then the oxidase activity is along the side. And this parental population was resistance to permethrin. And the mechanism that's selected for that mechanism was high levels of esterases, because they're the red is to the right of that kind of limits of susceptible population box. And when we took that resistant population and we further selected it with DDT, what we ended up doing is not selecting for more esterases in that colony, but we selected for oxidase overexpression as well. And so it's not a given that if you're switching from chemicals that you're going to get relief. And we did the same thing where we further selected them with some permethrin, and we continued really to increase that esterases, but with time we also increased the expression of oxidases. And then if we selected with malathion, so we switched chemical classes, we selected for higher levels of esterase. So we kept selecting, but we didn't really increase the oxidase activity in that population. And this is not, don't take home that, okay, if I have high levels of esterase in my population and I use malathion, I'm just going to make more esterases. The take home message here is that depending what chemical you switch to, you could affect your population in different ways. And it's unpredictable which way that it is going to go. And so just as a real world example of this is there was a West Nile virus outbreak in Maricopa County in 2010, and they were using a permethrin-based product. And so when we went in and they asked us to help, and during an outbreak, it's not the time to try and figure this out. Hopefully you have a surveillance program in place ahead of time. But when we looked at the permethrin resistance, it became clear to us that the mechanism behind that resistance to permethrin was mitigated by using DEF, which tells us it is an esterase mechanism. They had used malathion in the past, and they had resistance to malathion, so that was not going to be something that we could tell them use malathion instead of permethrin during this middle of this outbreak. But the mechanism giving them resistance for malathion was actually mitigated by using DEM, or diethyl malleate, which says that malathion resistance was based on glutathione S transferases. And when we actually measured those enzymes, we could see that it was mostly alpha esterases, beta esterases that were overexpressed. They really didn't have oxidase involvement, and their mosquitoes did have these overexpression of the glutathione S transferases, which was again resistance for malathion, and the esterases were resistance to the permethrin. And we did manage to get some cage field trials off on their two vectors. And you could see that this middle line in the first panel, Culex tarsalis, that's the highest label rate for that product. And they were only killing roughly half of the mosquitoes. In Quincke fasciatus, they were killing the mean was 81%. Now, because we knew it was an esterase mechanism, and I already mentioned this, there is one pyrethroid that is not an ester, it is an ether, and that is etofinprox. They switched to using etofinprox during this outbreak. And we were able to go back and do these cage field trials again, looking at the both the product they were using the previous year and the etofinprox product. And they had, in a single year, almost full recovery of their mosquitoes to exposure to permethrin by doing a wise selection of what to use next. And these are just the differences between them are just label rates, a mid-label rate versus a highest label rate. And so they could see that switching to the ether pyrethroid was beneficial, but if it had not been an esterase mechanism, this would not have worked. And when we look at the enzymes, and this is actually a couple years later, we can see that their alpha esterases are almost the same in their field populations as in their susceptible mosquitoes. And the same thing, even though we weren't doing this to manage for their malathion resistance, over time, their GSTs were also dropping out of the population. So just to give you, this is a chaotic system, and resistance is a wicked problem. But there are some general tendencies. If you don't have the ability to do all of this in-depth testing and figure out exact mechanisms, the general tendency is that if you're using organophosphates, they tend to select for esterases. Now, that was not the case in Maricopa County. So again, having concrete information and data trumps using a general tendency. Pyrethroids tend to select for overexpression of oxidases. Culex, as a group, tend to develop metabolic resistance over altered target site, but that does not mean that a population of Culex cannot develop an altered target site mechanism, and Aedes and Anopheles tend to develop the altered target site resistance over metabolic resistance. So there are some general tendencies, but every situation is going to be different. And the example I just gave you does not follow these trends, because malathion resistance was glutathione S transferase, and their permethrin resistance was an esterase. And so just to finalize and kind of pull all of this together, in a day-to-day surveillance program for insecticide resistance, knowing resistance is present, it really is the first step. But if you detect insecticide resistance, it is a very good idea, if you have the ability and something to strive toward building a program even stronger, is to come up with the ability to detect what the mechanism is, because knowledge of these mechanisms can be important in understanding the importance of that resistance on control. Again, if you have limited things you can switch to, but it's esterase and it's impacting on the age of the mosquito, maybe you can live with it a little bit better. Knowledge of that mechanism can help guide product choices across a landscape of multiple and cross resistance, because a program your surveillance should be looking at different sites within your program, and you should be monitoring those same sites over time. And resistance is not, once it's there, it's not set in stone. Um, you can do moderation. You can use the principles of IPM to minimize how often you have to go out and spray by using other tactics that would be part of an IPM program, and not spray when you don't have the mosquitoes present. All of this is part of integrated pest management, but unfortunately, there's no one size fits all for programs. There's no one strategy that I can sit here and say, oh, everybody, I gave you three strategies, but the one that works is the multiple attack. And so with that bad news, again, resistance can be managed and susceptibility can be regained if you're looking for it and you catch it early, and then you do have options. So I'm going to wrap it up, and I think we have 10 minutes for questions, and I'm going to turn it back over to Jennifer.
Thank you so much, Janet, for that very informative presentation. We have time now for just a few questions, so I'll kick it off with this first question. How can control programs account for local variation in resistance levels across different regions or even within a single city?
I think that across a region, certainly talk to your neighbors. If they're seeing an issue, that gives you maybe something to home in on, that, maybe I need to be looking for that as well. But as I showed in my one slide, even within a jurisdiction, the pressure for resistance is not just coming from public health. It comes from what homeowners do, what industry does, what golf courses do, what agriculture may abut where you are. And so you really have to develop a surveillance program where you are testing across your landscape. And, maybe when you start out, you can only test a few places, but it's always a capacity to try and build and improve on. And
So
sometimes
much.
it's trial and error. What do you do next?
Right.
And what worked on the north side of town may not work on the south side of town. And so you do have to dig in a little bit.
Definitely. Awesome. So we also have a question from the audience. Do you feel that using one treatment when finding mosquitoes, then switching to a different treatment at a follow-up visit, if they're still breeding, is beneficial in cutting down resistance?
So certainly using multiple modes of action makes it more difficult for resistance to develop. And we do have a larvicide product out there that has been used for decades and worldwide, and there's never been documentation of resistance developing to it, and that is BTI. Because BTI for mosquitoes, which is different than the BT used in agriculture for Lepidoptera and beetles, is a combination of 5 different toxins. And so the mosquitoes have never been able to develop resistance to all 5 of them in combination. But you can separate them out and you can artificially select resistance in the laboratory to the individual 5 toxins. and a couple combinations of them. So yes, it can be beneficial, and that's kind of the theory behind the multiple attack, is that you have different modes of action, and so you keep the population off balance.
Well, thank you, Janet. It doesn't look like we have any other questions, so just want to say, oh, last minute question.
Great.
Okay, one more. How or can changing spatial management schemes work on a much smaller scale, such as a housing apartment building, one apartment, or even one room?
So I think the example of that is kind of akin to what they do in malaria control, where you're treating a village to protect them from malaria. And so that strategy, which they had a very big trial in Mexico where they did this. They went in and they assigned, some houses got um, a residual spray with a organophosphate. Some of them got a residual spray with a pyrethroid. And they were able to actually, by doing this mosaic strategy, were able to regain some susceptibility in those mosquitoes. And it, like an apartment building would be maybe somewhat similar, in that if you have to treat all of the rooms in the apartment, that you could treat them with different products, with different active ingredients. But that sounds like that's not so much a mosquito question as maybe a cockroach or an ant question. And those products were, there's more active ingredients for those types of products. And they are usually, a lot of baits are being used for that as well. So I don't have a whole lot of experience with ants and roaches and those particular products, or even bed bugs, but the strategy could be maybe adopted. And it would take research to figure out if that was a good answer or not.
All right, so unfortunately, we're just about out of time. I see there's one more question that we won't be able to get to right now, but we'll be emailing the webinar recording as a follow-up, and we'll include Janet's response to that question and any others we receive in the next few minutes in that email. So just another huge thank you to Janet McAllister for the wonderful presentation. And now we just like to share, oh, sorry, Janet, can you stop sharing your screen?
Oh, yes.
Thank you. All right. So just as we wrap up, we'd like to remind attendees of an upcoming Grand Round seminar that is actually scheduled for later today. From bites to outbreaks, responding to the upsurge of dengue virus, which is scheduled at 3 p.m. Eastern Time later today. So this seminar brings together a panel of leading experts who will be providing comprehensive knowledge and actionable insights on dengue. So please make sure to register if you have not already. And as we wrap up, we just want to thank you so much for your time and encourage you to join us as a NEHA member if you aren't already. The more members we have, the stronger our voices and ability to address policy and funding at the national level. So thank you again to Janet for her presentation and to all of you for joining us today. And we hope you enjoy the rest of your day. So thank you very much.